Hello, everyone. Welcome to my video tutorial for Spatial Transcriptomics data analysis. In today's video tutorial, I'm going to show you how to use the non-negative matrix factorization method to perform dimensional reduction for spatial dataset. It becomes a very popular method to analyze spatial dataset. So NMF is a general method for dimensional reduction and feature extraction. So it can reveal signatures of distinct cell types in the spatial dataset, and also it can show spatial organization of tissue structures. So today we are going to use the STutility package. This package was built on top of Thread package, so it is easy to use if you know the Thread package very well. And also, the NMF dimensional reduction method was implemented in the package. Like we run PCA UMAP in the Thread package, we just need to run the NMF function. Then we can perform the NMF dimensional reduction for spatial dataset. So first we need to load the STutility package, tidyverse, and Arcana brewer. Once the packages are ready, we can start to prepare the spatial dataset. I'm going to use a 10 times Visium dataset for demonstration today. You can download any Visium dataset from the 10 times Genomics website. To load the data into STutility and perform the analysis, we need 4 data files. The first one is the filtered feature BC matrix H5 file. The second one is the tissue position CSV file. The third one is the scale factor JSON file. Then the last one is the image file. So you can see the advantage to use the STutility package to analyze spatial dataset. We can directly run the NMF method, and also we can load the original image. STutility package has a function called input from table to create a thread object from the 4 files here. So first we need to make a table for the files that we need to load the data. We get the sample data, the position file, the scale factor file, and also the image. Now we can make a table. You can see we have an information table contains the 4 files that we need to load the data. So next we can run the input from table function. At the same time, we can filter the data to keep the spots have minimum gene count bigger than 100 and minimum gene bigger than 5 and minimum total count for each spot. So the platform we are using is the 10 times Genomics Visium. So now we can load the data. Okay, you can see we created a thread object named as SE. Let's connect the data and have a look. You can see we have 13,760 genes, 2,702 spots in this dataset. In the Cs, we have the RNA data slot. So we loaded the expression data. We can have a look at the N features and the N count. First, let's make a corner scale, then we can use it for our analysis. So now we can look at the N features and the N count in this dataset. You can see we show the N features and the N count on each spot. So the corner scale, we set it from light gray to black, you can see the black dot has high M features and N count. So next we can note the image. As I said, this is one of the advantage compare the STutility package to Surat. We can note the original image. So let's use the node image function to node the image. Then we can use the image plot to have a look at the image. You can see in the plot window, we have the original image by H&E staining. You can see here we have the tissue image and also we have the information for the William slice. If you just want to see the tissue image, you can mask the image. We can run the mask image function. Then use the image plot. You set the type as masked. Let's run. You can see now we only have the tissue image. So we know that the expression matrix data for each spot, and also we know that the image. Now we can normalize the data. We are going to run the SC transform function from the Surat package. If we run the SC transform, we will normalize the data and also find the variable features. At the same time, we scale the data. So let's run SC transform. Okay, we finish the SC transform analysis. We can have a look at the Surat object again. You can see now in the assays, we have a SCT data slot. We can also use the view function to have a look at the metadata. You can see the row names and the cell names. We have the origin ident as the Surat object. We have the N count and the N features RNA, the ID for each spot. After SC transform analysis, we also have the N count SCT and the N feature SCT metadata. So after the data normalization and the data scaling, we can use the feature plot function to look at some gene expression on the image. We can run the features for the CCK and the MPP. Let's run. You can see we generated the plot for neurotransmitter CCK and also the myelin basic protein. You can see high MBP expression in those regions. In the mouse brain, they are the oligodendrocyte to express the MBP protein. So here we show the CCK and the MBP expression on the image created by each spot. We can also use the feature overlay function to see the CCK and the MBP expression on the original image. Here you need to set the type as raw, then we can see it on the original image. We can run. Now you can see we have the CCK and the MBP expression in each spot on the tissue, and also we overlaid the spots on the original image. So after the SCT transform analysis, we can run NMF to perform the dimensional reduction. So here you need to set a number for n factors because after the analysis, the spots on the tissue will be grouped into different factors. So the default setting for n factors is 20. For the demonstration, we are going to use 40, but you can try different factors to see what kind of result you can get from your dataset. So let's run NMF. To perform the dimensional reduction, this step is very similar to the STutility package like we run the PCA. So we performed the non-negative matrix factorization analysis. Because we set the m factor as 40, so we actually can use the dim plot to look at the factors from number 1 to number 40. So for demonstration, I'm going to show you, for example, the m factor 4 and 5. Let's run the ST dim plot. You can see now, we can see the spots in factor 4 and factor 5. They are in different anatomical regions in the mouse brain. We can also print a summary of the genes that contributes to the most dimensional reduction factor. Let's print the NMF factors. You can see here we printed the factor 1 to factor 5. You can see the M factors will be very similar to PCA. We have the positive genes and negative genes for each factor. You can also make a bar plot to summarize the topmost contributor genes for each factor. You can see for the factor 1, we have the AGT, SparkC, AppleE. We can use the factor 1 as an example to make the bar plot, which we need to use the factor gene loading plot function. Let's run. You can see we generated the bar plot for the most contributing genes for factor 1. So as I mentioned, in total we have 40 factors here. You can use the ST dim plot to look at each factors on the tissue and also print the top genes. So after NMF dimensional reduction, now we can cluster the cells. This will be the STutility function to find the neighbors. Here we just need to set the reduction as NMF. If we have a look at the STutility object now, you can see in the reduction we have the NMF method. So now we can find the neighbors. You can see we run the same method SCNN as the STutility package. Next, we can find the clusters. You can see with the default setting, we have 18 clusters. We can use the view function to have a look at the metadata again. You can see now we added the STutility clusters in the metadata. The default setting will be resolution 0.8. So we have the spots clusters. Then we can use some plot function to look at the gene expression in the STutility clusters. For example, we can use the wiring plot to look at the olig-1 and the TH expression, and we group the expression by STutility clusters. So the olig-1 will be highly expressed in oligodendrocyte. TH is a marker for dopaminergic neurons. Let's run. So you can see we generated a wiring plot for the olig-1 and the TH genes. You can run the same plot for the genes that you are interested for your research. So next we can look at the STutility clusters on the tissue. We can use the ST feature plot function again. So before that we can make a color vector to show the STutility clusters in different color. Let's make the color vector first. Now we can run the ST feature plot function. The features will be STutility clusters. Let's run. You can see we plotted the SWOT clusters on the image. So after the analysis, you can see the spots have been grouped in distinct anatomical regions in the mouse brain. You can see the different layers of the cortex, and the spots in the hippocampus, spots clusters in the midbrain. So here we plotted the SWOT clusters. But if you see recent publications, they are more interested in the M factors on the tissue image. Okay, so that's today's demonstration. I showed you how to perform non-negative matrix factorization dimension reduction method to analyze spatial transcriptomics data. You can see from recent publications, this method became very popular to analyze spatial transcriptomics data. I'm going to stop my demonstration today from here. You can install the STutility package and practice this method. Thank you for watching my video tutorials. See you next time.